This is a B6 video for the topic Inheritance, Variation and Evolution, and this time we're looking at genetic engineering. It can also be referred to as genetic modification, and we talk about GM crops, etc. But genetic engineering and genetic modification mean the same thing. Now, genetic engineering is about changing the DNA, the genome of a living thing to change its characteristics. It's important to realise that it's not about swapping a gene, it's just about adding an extra gene. Okay, so again, if you watch Animal Farm, you'll see that they have taken the fluorescent gene in a jellyfish and added that to the rabbit. Okay, so it's adding an extra gene, not swapping a gene. So, what kind of features might we want to genetically modify our crops for? We could go for pest resistance, frost resistance, disease resistance, herbicide, which is, so then you could spray weed killers and it would kill all the weeds, not the crops. So herbicide resistant, drought resistant, or longer shelf life. So if in the point of view of a farmer, it's really useful to genetically modify crops because they get a higher yield, therefore make more money and have a more successful harvest. Problem with genetically modified food is that if you are a farmer and you've genetically modified your crop and then next door the farmer prides himself on organic food, it's not actually possible then that will continue as organic food in the field next door, because you could get cross-pollination, and where does it stop? Where does that, if we want to stop genetically modified crops, how do we stop that? We'd have to destroy everything and start again, okay? Because we don't really know 100% that genetically modified food is okay to eat, so this is a major disadvantage. So how does genetic modification work? We've got quite a complicated picture here, but this is a classic one that the exam board like to talk about, and it's how we make insulin for diabetics. So we know that if you are type 1 diabetic, then you have your insulin injections, and this is how they create that insulin. So the first situation, or first part of it here, is that we have a cell from someone who can make insulin. Now this could be any cell, so the person won't know that, they won't feel anything happening. The cell is just removed, and then the chromosome with the gene on it is isolated. It's the gene that allows the cell to make insulin, okay, and that's cut out using enzymes. You can see just underneath there that individual gene is cut out of the DNA using enzymes, kind of like scissors. We think of enzymes as being like scissors, they chop things up. Okay, the next thing that happens then, we know that bacteria multiply really quickly. So we take a bacterium, we take some of the genetic information, one of the plasmid loops out of the bacterium. Again, we open it up using enzymes. The third step is that the gene that we cut out is then put into that plasmid from the bacteria. Okay, all of this done by enzymes. The plasmid is then put back into the bacterium. The bacterium is able to multiply rapidly, okay, roughly doubling every 20 minutes, and then all of those bacteria are able to make insulin. And here is the insulin, and the insulin is then harvested and given to type 1 diabetics. Okay, hope you found that useful. Thank you.